Hello everyone, myself Muskan Gupta and today we are going to learn about the topic photosynthesis. But before starting this presentation, I would like to tell you all that I have made some notes regarding this topic, so the link is in the description, so do check it out. It will be really helpful to you guys. So let's get started. So the topics we are going to cover today are: first, what is photosynthesis? Second, chlorophyll. Third, stomata. Fourth, main phases like light dependent and light independent. Fifth, adaptations by leaf. Sixth, factors affecting photosynthesis and the experiments related to it. Photosynthesis and its importance. What is photosynthesis? Photosynthesis is basically a process by which living plants make their own food, that is glucose and starch. From what? From carbon dioxide and water. By using what? By using the light energy. Now, what is the importance of photosynthesis? So the first thing is that food for all. Photosynthesis is ultimately the source of energy and food for all living beings. And the second thing is the oxygen to breathe in. Photosynthesis is the only biological process which releases oxygen into the atmosphere. So this was about the photosynthesis and its importance. So now let's see what is chlorophyll. Chloro means green and phyll means leaf. So it is the green pigment found in the plants and it is contained in microscopic cell organelles called chloroplast. So now what are chloroplasts? Chloroplasts are minute oval bodies bounded by a double membrane and their interior contains closely packed flatted sacs known as thylakoids. What are they? They are closely packed flatted sacs arranged in piles known as grana, lying in a colorless ground substance called what? They are called stroma. And the chloroplasts are mainly contained in a mesophyll cells located between the upper epidermis and the lower epidermis. There is also a very important thing about chlorophyll. That is, chlorophyll is highly sensitive to light. So too much light may destroy chlorophyll. So this was about chlorophyll and the chloroplast. Now let's see about stomata. What is stomata? Stomata are minute openings occurring in a large numbers on the lower surface of the leaves. So the opening of the stomata is done during the daytime. In daytime, ATP is produced due to photosynthesis. This ATP is utilized to pump potassium ions into the guard cells. Because the potassium ions are pumped into the guard cells, they become hypertonic. Then the endoosmosis occurs and the guard cells become turgid. And because the guard cells become turgid, the stoma opens. Now let's see about the closing of stomata. The photosynthesis stops during the night time. The ATP is utilized and the efflux of potassium ions from guard cells is done. The guard cells becomes hypotonic. Then the exoosmosis occurs and the guard cells becomes flaccid. Then the stoma closes. So this was about the closing and the opening of stomata. Now let's see about the main phases. There are two main phases. First, the light dependent phase and second, the light independent phase. So first we learn about the light dependent phase. The light reaction occurs in this phase have two main steps. First, the activation of chlorophyll. The chlorophyll on exposure to the light energy becomes activated by absorbing photons. Second, splitting of water. The absorbed energy is used in splitting of water molecule, that is hydrogen and oxygen, and releasing electrons. So the reaction for this is 2H2O gives you energy of 4 photons, 4H plus 4E minus plus O2. This reaction is also known as photolysis. But now, what is photolysis? Photolysis occurs in the gland of chloroplast and is defined as the splitting of H2O molecules in the presence of light energy. Now let's see about light independent phase. The old term was dark phase, and that term did not mean that it occurs when it is dark. That means at night. It only meant that this reaction are not dependent on light. That's why it is now better to call light independent phase. So in this phase, the conversion of glucose into starch and other chemicals take place. Several glucose molecules are transformed to produce one molecule of starch. This process is also known as polymerization. The process of conversion of many simpler and smaller molecules into a complex, bigger molecule is termed as polymerization. So, this was about the main phases of photosynthesis. Now, let's see about the adaptation in leaves to perform the process of photosynthesis. First, large surface area. Large surface area for maximum light absorption. Narrowing of leaf in certain plants is to minimize transpiration in order to conserve water, like in pine trees. Second, leaf arrangement. Leaf arrangement at the right angle to the light source to obtain maximum light. Third, cuticle and the upper epidermis. The cuticle and the upper epidermis are transparent and waterproof to allow the light to enter freely. Fourth, numerous stomata. Numerous stomata allow rapid exchange of gases like oxygen and carbon dioxide. Fifth, the thinness of leaves. The thinness of leaf reduces the distance between the cell and the rapid transport. Sixth, the chloroplast. The chloroplasts are concentrated in the upper layers of the leaf to obtain light energy quickly. Seventh, extensive vein system. Extensive vein system is for rapid transport to and from the mesophyll cells. Now, what are the end results of the product of photosynthesis? There are three end products. First, glucose, that is C6H12O6. Second, water. And third, oxygen. This was about the adaptation and end result of photosynthesis. Now let's see what are the factors that affect photosynthesis. There are external factors and the internal factors. The external factor contains light intensity, carbon dioxide concentration, temperature, and water content. Whereas internal factors contain chlorophyll, protoplasm, and the structure of leaf. These are the factors that affect the process of photosynthesis. Now let's see the experiments on photosynthesis. First, iodine test. To test a leaf for starch. Step 1. Dip the leaf in boiling water for a minute to kill the cells. Step 2. Boil the leaf in methylated spirit over a water bath till it becomes pale white due to the removal of chlorophyll. The leaf now becomes hard and brittle. Place it again in hot water to soften it. Now spread the leaf in a dish and pour iodine solution on it. The presence of starch will be indicated by a blue-black color. A leaf without starch will be shown in brown color. Now let's move on to the experiment 1 to show that chlorophyll is necessary for photosynthesis. First, we'll take a plant which has leaves that has some green and some non-green areas. We'll destarch the leaf by keeping the plant in a dark room for few days. Now we'll place the plant in the sun. After few hours, we'll pluck one leaf and make its outline on a paper and mark the green and non-green areas on the outline. Now we'll test the leaf for starch. only the green parts of the leaf will turn bluish, showing the presence of starch. Now let's move on to the experiment 2 to show that sunlight is necessary for the process of photosynthesis. Step 1, we'll take a plant with destarched leaves. Then we'll cover one of its leaves with black paper. Then we'll place the plant in the sun. After few hours, we'll test the leaf which is covered by the black paper for the presence of starch. It will be observed that the only parts that were uncovered will turn black and blue, showing the presence of starch. Now let's see about the experiment number 3 to show that carbon dioxide is necessary for photosynthesis. We'll take a plant with destarched leaf or a leaf which is destarched. We'll insert half of the leaf in a conical flask that contains potassium hydroxide. Now, why potassium hydroxide? Because it absorbs the carbon dioxide. Now, we leave the leaf in sunlight for few hours. After few hours, we'll test the leaf for starch. The part of the leaf which was exposed to the atmospheric air becomes blue-black, and the part of the leaf inside the flask containing potassium hydroxide does not become blue and black, showing that carbon dioxide is necessary for photosynthesis. Now let's see the experiment number 4 to show that oxygen is produced during photosynthesis. For this experiment, we'll take a water plant like hydrilla. Place some water plants in a beaker containing pond water and cover them by a short-stemmed funnel. Then we'll invert a test tube full of water over the stem of the funnel. Then we'll place the apparatus in the sun for few hours, and the bubbles of the gas will be collected in the test tube. So, if a glowing splinter bursts into flame, which shows the presence of oxygen, thus we conclude that oxygen is produced during the process of photosynthesis. So guys, if you have any questions regarding this topic, then please feel free to comment those questions in the comment box, and I will try to answer them as soon as possible. So let's here conclude this chapter, and thank you so much for watching this video, and do subscribe to my channel for more videos like this. So, and yeah, don't forget to refer those notes. The link is in the description, so do check it out. Thank you so much.